Hyaluronic acid and cartilage regeneration.
Hyaluronic acid is a glycosaminoglycan, a naturally occurring polysaccharide that is abundant in the extracellular matrix of connective tissue, synovial, joint fluid, hyaline, cartilage, and vitreous of eye, and skin dermis. In humans, it serves as the ground substance of dermis, fascia, and most fluid mediums because of its viscoelastic properties. It is found in all vertebrate animals and is biofilm around bacteria. Hyaluronic acids have specific physical and biochemical properties in normal tissue that make them ideal structural compounds. When incorporated into aqueous solution, hydrogen bonding occurs between adjacent carboxyl and N-acetyl groups. This feature allows hyaluronic acid to maintain conformational stiffness. It also contains numerous repelling anionic groups which bind cations of water molecules, which allows it to retain water. Approximately 1 gram of hyaluronic acid can bind up to 6 liters of water. Hyaluronic acid structure consists of disaccharide units of D-glucuronic and D-acetylglucosamine connected by alternating beta-1,4 and beta-1,3 bonds. One of the first measurable signs of skin aging occurs with changes in mechanical properties of skin. Hyaluronic-based fillers have been primarily used for skin rejuvenation, such as visible wrinkles and line fillings. Hyaluronic acid is a biodegradable, biocompatible, and non-immunogenic. Products were utilized for 3D volumization and compensating for sleep-associated volume loss. Hyaluronic acid-based fillers brought about the discovery of improved mechanical properties of the skin following field implantation and persistence of results after retreatment. Restorations of extracellular scaffolds stimulating formation of younger components of the skin by injectable cross-linked hyaluronic acid. Enhanced structural support of extracellular matrix by injected hyaluronic acid is found to increase keratinocyte and fibroblast proliferation as well, resulting in epidermal thickening and expenditure of dermal vasculature. For an effective dermal filler, cross-linking native hyaluronic acid forms larger, more stable molecules with longer residue time in tissues. The bonding creates a larger macromolecule, transforming the hyaline fluid to a more cohesive gel. The gel is hygroscopic, meaning it swells by absorbing water from the surrounding atmosphere. Particulate cross-linking thus creates water-insoluble gel that will remain stable in tissue as they are slowly reabsorbed over a period of months. Natural hyaluronic acid has a half-life in tissue of 1 to 2 days before undergoing aqueous dilution and enzyme degradation in the liver to carbon dioxide and water. As one ages, continuous exposure to free radicals inflicts damage to the skin. The complex process of aging is affected by both genetic and extrinsic factors. Most important structural components of the dermis are collagen, elastin, and ground substances, all produced mainly by fibroblasts. For successful production of connective tissue components, fibroblasts need stable collagen scaffold which they can bind. This binding is carried out through specific receptors on the fibroblast surface called integrins. Integrins attach to molecules of collagen in the extracellular matrix and bridge them with tubular cytoskeleton inside the cell. Intracellular microtubules pull fibroblast skeleton inward, pull adhesions with extracellular stable matrix, and push the cells to spread. This results in increased mechanical tension inside the fibroblast and stimulates production of new collagen and downregulation of collagen-degrading enzymes. Fragmentation of the scaffold causes fibroblasts to collapse and reduces their procollagen function. Impaired fibroblast function with reduced collagen synthesis translates into atrophy and wrinkling of aged skin.
As it was mentioned previously, hyaluronic acid is a substance naturally present in the human body, and it is found in the highest concentrations in fluids in the eye and the joints. Because hyaluronic acid is found in joints in such large quantities, hyaluronic acid plays a major role in helping and enhancing cartilage formation. In articular cartilage, hyaluronic acid is present as a coat around each cell or chondrocyte. Therefore, it functions as a lubricant and permits the sliding between adjacent tissue layers. Hyaluronic acid is also involved in many cellular processes, including but not limited to cell signaling, cell differentiation, and matrix deposition. Hyaluronic acid contributes to the structure and function of tissue microenvironment by working as a molecular powerhouse with critical roles in homeostasis, pathological disease onset, progression, and recovery or decline. In addition, hyaluronic acid plays a significant role in the nature of its interactions with the extracellular matrix subsurface receptors, including its major receptor CD44. Because of its large water of hydration, hyaluronic acid also provides a matrix that enhances cell migration. Additionally, hyaluronic acid regulates cell behavior during embryonic development. It regulates healing processes and inflammation and tumor development. Data suggests that hyaluronic acid production by cumulus cells during maturation is essential not only for cumulus cell expansion, but also for oocyte maturation and further embryo development. It has also been proven that hyaluronic acid modulates through specific hyaluronic acid receptors inflammation, cellular migration, and angiogenesis, which are the main phases of wound healing. Hyaluronic acid, or more specifically sodium hyaluronate, has shown to enhance both tumor growth and CD44 expression of cancer cells at wound sites, suggesting that the use of sodium hyaluronate might stimulate the growth of residual tumor cells. Because of hyaluronic acid's physical and healing properties, it has been widely used in the production of supplements, lotions, and eye drops to provide structure and moisture to damaged tissue. Based on this information and numerous studies published previously, a group of scientists from Stanford University in California published an article called Elastin-like protein hyaluronic acid hydrogels with decoupled mechanical and biochemical cues for cartilage regeneration. Hydrogels are highly absorbent networks of polymer chains that are hydrophilic and are sometimes found as colloidal gel in which water is the dispersion medium. In fact, an example of a hydrogel-forming natural polymer is cellulose, which we discussed in class recently. Now, going back to the research article, previous studies actually show the promise of hyaluronic acid-based hydrogels in supporting cell-based cartilage formation, and varying hyaluronic acid concentrations generally leading to simultaneous changes in both biochemical cues and stiffness. Although it is still unknown how cells respond to the change of biochemical content of hyaluronic acid, the researchers reported an adaptable elastic-like protein hyaluronic acid hydrogel platform using dynamic covalent chemistry, which basically means that the chemists used a synthetic strategy in order to make a complex supramolecular assembly from discrete molecular building blocks. In order to create the elastin-like protein hyaluronic acid hydrogels, the ELPHA hydrogels were created through dynamic hydrazone bonds through the reaction between hydrazine-modified ELP and aldehyde-modified hyaluronic acid. The ratio of aldehyde groups to hydrazine groups was maintained constant, and hydrogels had variable hyaluronic acid concentrations of 1.5%, 3%, and 5%. The purpose of this research study was centered on evaluating the effects of hyaluronic acid concentration on cell-based cartilage regeneration. The experiment was carried out by first dissecting articular cartilage from the femoral patellar groove of both stiffle joints from a 3-year-old calf. From this dissection, neonatal chondrocytes were then isolated and encapsulated within ELPHA hydrogels with the corresponding hyaluronic acid concentrations of 1.5%, 3%, and 5%. After 7 days in culture, the total RNA from all 3 groups of hydrogels was extracted from chondrocytes in order to evaluate gene expression levels. After 3 weeks of culture, all 3 groups of ELP-HA hydrogels were digested using a solution rich in cysteine protease enzyme, and from this digestion, DNA content was quantified and measured. Sulfated glycosaminoglycan was measured by using DMMB, which stands for 1,9-dimethylmethylene blue, and total collagen was quantified through an acid hydrolysis reaction. Lastly, in order to visualize the new cartilage extracellular matrix production and distribution, a technique of immunofluorescence staining was performed after 21 days in culture. Once the testing procedure was completed, the researchers focused on analyzing the levels of aggregan, SRY, and type 2 collagen. Aggregan is a proteoglycan core protein, and it's also known as chondroitin sulfate proteoglycan. SRY stands for sex-determining region Y, and it's a DNA-binding protein also known as gene regulatory protein, encoded by the SRY gene that is responsible for the initiation of male sex determination in humans. And collagen, which is the most abundant protein in the human body, and it's found in muscles, bones, skin, blood vessels, the digestive system, and tendons. Collagen is what helps give our skin elasticity and strength. Based on the observations of this study, the researchers found out that increasing hyaluronic acid levels led to a dose-dependent increase in gene expression of all tested cartilage markers by chondrocytes as shown in the red highlighted regions. Similarly, increased hyaluronic acid levels suggested a positive role in stimulating cartilage-forming capacity of chondrocytes and a more intense sulfated glycosaminoglycan deposition. This can be seen by comparing images A and C and images D and F. However, not all of the findings implied a positive result. In fact, the increase of hyaluronic acid levels to 5% actually expressed a decrease in cell proliferation to 5-fold. This is represented by the 5% column in image A. So what are the implications of this study? Based on the numerous research studies on hyaluronic acid and its healing properties, pharmaceutical companies have come up with different ways of synthesizing it, including the extraction of hyaluronic acid from roosters or simply by utilizing bacteria to culture it in the lab. For this reason, hyaluronic acid is widely used in the medical field for osteoarthritis treatment, corneal transplantation, and other eye procedures, lip and nose fillers and cosmetic procedures, healing of wounds, burns, skin ulcers, and even in order to prevent the effects of aging. Hyaluronic acid continues to be a topic of interest, and most of its current research is being dedicated to hyaluronic acid bioconjugates, hyaluronic acid streptococcal fermentation, and hyaluronic acid in aging.